So it's live.
Yep.
And
You're ready to go.
Say something. My sound just went weird.
Okay, so hello everybody. Welcome to the Kids First program spring webinar. We're going to give 1 minute for people to join, and we will be starting in 1 minute. I'm glad to see many of you already here today. Thank you for sharing your time and attending today's spring webinar. We're happy to share some of the developments and news going on in the Kids First program. I think we already hit 2 o'clock, so I will start our program for today. Again, welcome to the Kids First program webinar. And thank you for sharing your time with us today. In a nutshell, the Kids First program is developing a large cloud-based data resource to support research and new discoveries in pediatric research, specifically pediatric cancer and structural birth defects. We expect that the research may lead to new developments in prevention, diagnostics, and treatments to help patients and families. We have a full agenda today with highlighting Kids First program research, tools development, and pilot announcement. We have an announcement today on a pilot that the Kids First program is running. And I think you will all be interested in that, as well as mechanism of support of research using Kids First data and resources. I'd like to ask you guys, if you have questions, please use the chat box, and we will be addressing all the questions in the end of the program. I will start with introduction about myself. I'm Marcia Fournier. I am a cancer researcher and entrepreneur. I have been working on genomics and precision medicine for the past 20 plus years. Specifically, I have been interested in how to use data to answer scientific questions and inform clinical decisions. I recently joined the Kids First program as a program manager, and I will be giving you an introduction about the program. Well, let's start with a little bit of the history of the program. The program came together with a combination of cancer awareness and a scientific hypothesis. Gabriella Miller was 10 years old when she died from brain cancer. But before that, she was a fearless advocate and fundraiser for pediatric cancer. And she left a legacy. The Kids First program is part of Gabriella Miller's legacy through a bipartisan bill signed by Congress in 2014, authorizing $12.6 million per year for 10 years to support pediatric cancer and other pediatric conditions research at NIH. And in 2015, the Kids First program was born with the first annual appropriation, and the program is funded through 2024. A little bit about the scientific hypothesis behind the Kids First program. The intersection of human development and cancer is not a new concept. And NIH has been interested in that intersection for a long time. I found, for instance, a workshop hosted by NIH in 2012 addressing birth defects and cancer. Also, many reports in the literature have been showing shared mutations between pediatric cancer and birth defects, such as clinical oncology drug targets BRAF, MAPK, and ALK. But not only very recently, some studies, large-scale studies, supported the association between birth defects and increased cancer risk. And this study I want to point out, authored by one of our investigators at Kids First program, Dr. Philip Lupo, one of our speakers today, with a study including 10 million births, showing that association of birth defects to increased cancer risk that also suggests to persist into adulthood, as highlighted by the New York Times recently. So who is behind the Kids First program? The Kids First program is an NIH Common Fund program that is overseen by a working group formed by multiple institutes inside the NIH. And our main goal is to accelerate collaborative research and promote new discoveries. How we are doing that? The Kids First program is developing a large data resource in the cloud to house fair data, findable, accessible, interoperable, and reusable data, promote data sharing, data analysis, and data visualization, development of tools, and foster collaborative research. When we ask our investigators what differentiates the Kids First program from other data platforms, we hear that, one, the program offers the opportunity of whole genome sequence. That's right. The program is actively investing on generating data, and I will talk more about that soon. Also, the data is publicly available, but you're not going to find out only the sequencing or transcriptome data. This data is associated with phenotypes and clinical data, which enhance the meaning of this data and also give additional opportunities of data analysis. And third, the Kids First program data resource brings multiple pediatric conditions and childhood cancer data, which also offers the opportunity of rich analysis, for instance, looking at commonalities and difference between phenotypes. Now, how the Kids First program supports research. So one of the main mechanisms the NIH, we at Kids program have been using to support data generation is the EXO1 mechanism. And if you are interested, go to grants.nih.gov. And we've been using this mechanism to support data generation, as I said, through selected projects that are selected through the NIH peer-reviewed process and portfolio analysis. So select investigators send DNA and RNA sample to the Kids First program sequence centers. The Broad Institute, Hudson Alpha, and St. Jude are the Kids First program sequencing centers. The sequencing centers then generate the genomic data with the investigator samples, and this data goes to a data resource center. The Kids First Data Resource Center then compile the sequencing data with phenotypes and clinical data provided by the investigators and give that back to the investigators. And investigators have 6 months exclusivity to work with the data before the data is made public. The data is made public, and the good news is through the Kids First Data Resource Center portal, this data is available not only for the EXO1 investigators, but also to researchers nationally and internationally. So it's a public data. We bring harmonized data, analysis and visualization tools and resources in the cloud with already over 3,000 registers used since its launch in 2018. just an idea of the size of our data resources. We already have almost 30,000 subjects as part of our data resource. So to join the Data Resource Center, please visit the portal. You're going to hear more about that today as part of our agenda for the spring webinar. And so the Kids First data sharing is fast growing. We observe about 20% growth since fall 2021. So from the 56 projects we supported for sequencing, 24 studies are already available at dbGaP. So if you are interested in getting access to the Kids First program data, please visit our dbGaP website. It's here, shown here, but also maybe someone can put in the chat box. Through the dbGaP, you can get access to this data. And recently, since the fall 2021, we've released three new projects, and I will show you a little bit of the flavor of the new projects, bringing almost 4,000 new subjects to the data resource. And we expect new data will be released to the public very soon, so stay tuned to the upcoming data release announcements that the Kids First program will make. Now, this is how the program supports data generation. We also have mechanisms that will support the data analysis. After researchers has the data, they can apply to multiple NIH mechanisms, and you're going to learn more about the mechanism with today at the webinar. My colleague James Coulombe will talk about that. And I want to highlight specifically an opportunity that is open with a due date on June 10, where we are calling for projects that can enhance the use and the data that we have at the Kids First program through tool development, data harmonization, and data analysis projects. So this gives you a little, an example of multiple NIH support mechanisms using Kids First data and resources. So we found over 70 NIH research grants in the period of 2016 to 2021. And most of them are research grants, R03 and R01s, but also we were pleased to see other mechanisms being using the Kids First program data resource, including career development and fellowship awards. We feel humbled to be participating on forming the next generation of pediatric cancer researchers. What is the future for the program? So we focus on the future on gathering additional data on the childhood cancer and structural birth defects field, and multiple data types, adding to the genomic and transcriptomic data that we are already generating, start generating epigenomic and proteomic data. We would like to continue development and improvement of our data resource to enable discovery and data generation, cross-disease collaborations, and interoperability. And you're going to hear more about that today when you hear from Valerie Cotton. And we also aim to invest in expert-driven activities, again, looking for projects that are developing new tools, data harmonization, and data analysis tools. The Kids First program is also part of a larger data ecosystem, collaborating and developing interoperability with multiple data platforms inside NIH and outside NIH. I would like to mention specifically the NIH cloud-based platform interoperability program, which Kids First is collaborating with the Biodata Catalyst, Anvil Project, and Cancer Research Data Commons. And you're also going to hear more about that today through our agenda. And with that, I will thank you. And again, if you have questions, feel free to add your questions to the chat box. And I'm going to call our keynote speaker to the podium, Dr. Philip Lupo. He will deliver the keynote on cancer risk in children with birth defects, how Kids First can move observation into understanding. Dr. Philip Lupo is the director of the Epidemiology and Population Science Program in the Texas Children Cancer and Hematology Center. He is a Kids First principal investigator, and his area of research is cancer risk in children with birth defects. Dr. Philip Lupo, I would love to invite you to join me and share your experiences.
Thank you very much, Marcia. It was quite an introduction, as well as I really enjoyed hearing your background setting up the story of Kids First. So bear with me while I pull up my screen. And I trust you can see everything okay. All right. So again, thanks again for that introduction, Marcia. I am really honored and privileged to talk a bit about our research and how Kids First has really moved that area of study forward over the past few years. So I was actually at that meeting in, excuse me, 2012 that the NIH sponsored, trying to address this question of cancer risk in children with birth defects. And that really set the stage for a lot of the work that I'm going to be talking about today. And just to get started in terms of an outline, I'm going to share some of the observations that we've been able to generate through our registry-based studies, which I think has really set the stage for the sequencing projects that have been selected as Kids First cohorts. And I'm actually going to highlight two of those studies that have been selected for X01 funding through Kids First. The first of which is our Go Back, or genetic overlap between anomalies and cancer in kids study, where we're really trying to identify novel congenital anomaly cancer predisposition syndromes. And the second study that I'm going to highlight is our Down syndrome acute lymphoblastic leukemia study, which I think will hopefully point to the utility of Kids First in identifying those children with congenital anomalies who are more or less likely to develop cancer. So as Marcia said at the outset, I think there's actually quite a bit that we know about birth defects and cancer risk. And I've been using this illustration based on a March of Dimes categorization of birth defects for the last few years, but I think it's really important. So, I mean, obviously, when we think about birth defects, you can categorize them in multiple ways. And I find this designation by the March of Dimes to be particularly useful in thinking about cancer risk. So one of the most important groups of birth defects that we think about are those that are driven by chromosomal anomalies. And again, we do know through decades of evidence that children with certain chromosomal anomalies are more likely to develop cancer. And probably one of the best examples or best known examples of that would be trisomy 21 and acute lymphoblastic leukemia or acute myeloid leukemia. There's also a group of defects that are known to be due to single gene defects or Mendelian syndromes. And again, I think a good example of that would be something like Costello syndrome, which is characterized by dysmorphic features, congenital heart disease, and other birth defects, and the subsequent risk of certain malignancies like rhabdomyosarcoma. But really, the largest grouping of birth defects really fall into this multifactorial or what some people call non-syndromic group of birth defects. And I think this is where we know less about cancer risk, or at least we did know less about cancer risk, but where studies over the last decade have really moved our understanding. So what are some research questions related to this, and how can Kids First improve our understanding? So obviously, one of the most important questions, I think, in terms of understanding biology and thinking about surveillance and prevention would be, what are the birth defects that are associated with specific cancers? And then secondly, could some of these associations represent undiscovered Mendelian syndromes? And then importantly, why do some children with birth defects develop cancer while others do not? And can we leverage this information again to think about treatment and or surveillance strategies for these individuals? So that really led to a multi-state project that is actually ongoing still, but started with 4 states, as you can see here from this slide, including Texas, Michigan, and Arkansas, as well as North Carolina, where we linked birth defects registries with cancer registries in these locales to generate a birth cohort of a little over 10 million children. And with that cohort, we were able to identify 4 groups of individuals where we could really dig into cancer risk among children with and without birth defects. And I'd like to point out Jeremy Schraw, who was originally a postdoc in my group, but is now an assistant professor here at Baylor College of Medicine. Did a great deal of work in harmonizing and assembling these data for analyses. And again, the 4 important groups are outlined here in these boxes, but you can see even with a cohort of 10 million individuals that we had a little over 2,000 children who were born with a congenital anomaly or a birth defect who went on to develop cancer. So one of the largest assessments of its kind. And some of the 10,000-foot observations from that analysis were consistent with some smaller and older reports, but I think notable nonetheless, and as you can see, relatively precise with our sample size. So what we observed is that children with non-chromosomal birth defects were about 2.6 times more likely to develop cancer compared to children without those birth defects. Again, this is pretty consistent with previous studies evaluating non-chromosomal birth defects and cancer risk. And then children with chromosomal anomalies were about 12 times more likely to develop cancer compared to children without such chromosomal anomalies. And then moving on to specific birth defects and specific cancers, we did note some interesting observations that I think will lead to, I hope, findings from the Kids First program and populations that have been included as part of Kids First, which I think could really be explored to understand these associations. So the first of which is that we noticed embryonal tumors were associated with a range of birth defects. And this was particularly true for hepatoblastoma and neuroblastoma. And again, you'll know that neuroblastoma is included as one of the Kids First cohorts. Secondly, we saw the same pattern of associations across germ cell tumors, another cohort that's been included as part of Kids First sequencing. And then moving from specific cancers to specific birth defects, we did notice that children born with CNS defects were at an elevated risk of developing certain brain tumors, which I think is particularly interesting if you think about common developmental pathways that might have been disrupted at key points during gestation. Secondarily, we noticed that children with oral clefts actually did not have the same cancer risk compared to children with other defects. And I think this is an area worth exploring, especially in light of the number of oral cleft cohorts that have been included as part of Kids First. And then digging down deeper into those seemingly non-syndromic birth defects that were associated with cancer, again, because of our sample size, we were able to look at a number of specific associations that had previously been excluded in analyses that were published. And notably, among those 72 associations, 40 were significant after correcting for multiple comparisons. So I think this points to the importance of birth defects and cancer risk and the role of disrupted development on cancer in children. So a few specific associations among children with seemingly non-syndromic birth defects that I'd like to point out are listed here. Specifically, children with certain congenital heart defects were more likely to go on to develop certain embryonal tumors like hepatoblastoma and neuroblastoma. Again, I think this points to the utility of Kids First in that there are cohorts that have been sequenced of children both with congenital heart disease as well as neuroblastoma. So thinking about the common pathways that underlie these phenotypes, I think is a rich area to explore with those existing data. And then there were other associations that hadn't been previously reported that we were particularly interested in. And I'm going to draw your attention to the association between choanal atresia, which is a type of craniofacial defect, and acute leukemia. So in this case, children with choanal atresia were about 10 times more likely to develop acute leukemia, which was particularly interesting to us because it wasn't an observation that had been made, at least one that we noticed in the literature, and not something that's associated with classical cancer predisposition syndromes. The other observation from the study, and I think this points to the challenge in trying to call birth defects non-syndromic, is that we noticed with increasing number of seemingly non-syndromic birth defects that the risk of cancer increased as well. So you can see here from this cumulative incidence plot that with each increasing number of major non-syndromic birth defects, the risk of cancer increased significantly, whereby children with 4 or more defects were about 6 times more likely to develop childhood cancer compared to their peers without birth defects. And again, these are children that did not have a syndromic diagnosis, at least in the birth defects registry data that we had access to. And again, what I think this points to is likely that there are undiscovered syndromes within these data that could yield insights about cancer predisposition and about development in general. So moving on from those registry observations into trying to understand what's driving these associations has really been the impetus of the enrollment portion of our study, or the Go Back Family cohort. And Dr. Sharon Plon and Dr. Annika Sabo have been amazing partners with me in this endeavor. And aside from the work that we've done in linking those registries, we've also used those registries to identify families where the proband has both a congenital anomaly as well as a cancer diagnosis to conduct whole genome sequencing, both on those individuals as well as family members. And I'm going to highlight one family in particular here that I think is indicative of how we can use Kids First to really get a better sense of what's driving these associations and what might be important for thinking about novel syndromes. So in this one family, as you can see here, which has really been led by Samia Sasudia, who's a PhD student in Dr. Plon's lab, we observed about a 5 kilobase exon deletion in USP9X. And this is obviously not present in either parent or the proband's sister. And this was validated in a PCR experiment. But what's interesting about the phenotype of this proband is that it really matches nicely, I think, with the registry observations that we had in the Go Back study. So for instance, the child had multiple structural birth defects and did not have a syndromic diagnosis. If you were to look at this set of birth defects, you might imagine that the child had Charge syndrome, for instance, but did not have a CHD7 variant. And also the child had choanal atresia and diagnosis of precursor cell lymphoblastic leukemia. So this association between choanal atresia and acute lymphoblastic leukemia that we saw in our registry data, which led us to enroll this particular family, I think really bore out, I think, some interesting observations in terms of genomics. Because again, remember, this association that we observed was not necessarily one reported previously. And then I think digging into USP9X and its role in both development and cancer, I think points to some of the evidence that Marcia alluded to at the beginning of her presentation, is that these genes really can be implicated in several developmental and cancer pathways. And this is true for USP9X, as you can see from this biorender illustration that Samia prepared. And then Samia has done a great deal of work to identify families that are characterized by these USP9X variants. And to date, there have been 42 identified individuals with variants in USP9X. And they all seem to be associated with a female-specific syndrome that includes both developmental delay and congenital anomalies. And what's even more interesting among these 42 individuals with this novel syndrome is that 3 of them have gone on to develop acute lymphoblastic leukemia. So if we were to look at ALL risk in our USP9X cohort, or in this USP9X cohort versus SEER data, you can see there's a significant enrichment in leukemia incidence and risk. Which I think is notable in that this is not, again, a known or a well-characterized leukemia predisposition syndrome. And then if you move from germline sequencing to somatic sequencing of tumors from this pan cancer analysis that was published several years ago now in Nature by Ma et al., one of the most mutated genes in the context of acute lymphoblastic leukemia somatically was USP9X. So I think this is beginning to lead to a hypothesis that I hope you're drawing, at least through the presentation of these data, that USP9X could be a novel ALL susceptibility gene that's associated with a charge-like syndrome. So again, I think this is, I hope, an example where we move from association in our registry-based studies to genomic sequencing of informative families to identify these alterations that could underlie novel syndromes. So what are our next steps related to this? Again, I mentioned that our Go Back cohort was selected for sequencing as part of Kids First. So we're really hoping now to move beyond, again, these registry-based studies to enroll more families where these children have both a birth defect and a diagnosis of cancer. And through our X01, we're planning to determine the frequency of known cancer predisposition variants among children with congenital anomalies and cancer, and then to identify novel variants that could underlie syndromes that haven't been characterized, like with our USP9X example, and also describe the landscape of somatic alterations in these children. And again, this is an effort that Sharon Plon and Logan Spector and I have been leading over the last 4 years. And it builds largely from the Children's Oncology Group. And for those of you who aren't familiar with COG, it's an amazing resource, not only for developing clinical trials for children with cancer, but also for observational studies in these populations. So you can see there are more than 200 research sites throughout the United States that are affiliated with the Children's Oncology Group. And about 90% of children diagnosed with cancer in the US are cared for at a COG member site. And because we can leverage COG protocols like Project Every Child or APEC 14B1 to enroll patients into our studies, it gives an even richer platform to study cancer risk in these individuals. And an added component to Project Every Child is a recent questionnaire that was developed and implemented that does ascertain birth defects or structural birth defects at the time of diagnosis. And as you can see here, it's a limited list, but one nonetheless that we can use to identify children enrolled in Project Every Child that have a reported birth defect. And you should know that there are currently 25,000 children that have been recruited to Project Every Child. So again, you can imagine the power behind not only consent to this protocol, but the biological specimen banking that goes along as part of it. And our X01 is really going to focus on all children that are part of Project Every Child that have a malignancy as well as a reported birth defect, as you can see here in this table from our grant application. And as part of that effort, we're going to be sequencing a little over 700 individuals, and that's going to include both their germline and tumor samples that will be included for whole genome sequencing, and additional exome and transcriptome sequencing on a subset of the tumors that are banked as part of Project Every Child. But as you can imagine, from Project Every Child, this list is very limited in its scope. And one thing that we've been able to build from are the other mechanisms that are part of Kids First, specifically in the RO3 mechanisms to enhance phenotypic efforts in Kids First cohorts. So because this list is relatively limited, we recently obtained RO3 funding to collect extensive phenotypic and clinical data from children with congenital anomalies and cancer that are enrolled in Project Every Child. And then we're working closely with Allison Heath and Adam Resnick to integrate this information into the Kids First Data Resource Center. And I think this is going to be a powerful tool to expand upwards as we think of integrating data from Project Every Child into the DRC, which I think is a goal that a lot of us would like to see come to fruition. So overall, in terms of go back, I think, I hope I've showed that these population-based registries can really be leveraged to inform novel genomic analyses. And what's important, I think, is that birth defects based on these estimates could account for upwards of 10% of childhood cancer. So clearly an important consideration in thinking about the etiologies of these conditions. Of course, the reasons for these associations are complex and probably involve multiple pathways, but I do think in some children, inherited and de novo genetic variants are likely to explain the co-occurrence of both phenotypes. So moving on from our work in Go Back to Down syndrome associated leukemia in particular, I think most of us know, again, that trisomy 21 is one of the strongest predictors of acute lymphoblastic leukemia, as well as AML. In fact, we've known this for decades. And about 2% of children with ALL and about 10% of individuals with childhood AML have a diagnosis of Down syndrome. But what's also interesting in relation to Down syndrome associated acute lymphoblastic leukemia is that the clinical features are relatively different than among those without Down syndrome. So we see distinctive genetic alterations in the leukemia profile of children diagnosed with Down syndrome, whereby those typical ALL alterations like hyperdiploidy and ETV6-RUNX1 are less common, whereas CRLF2 and JAK alterations are much more common. So about 50% of children with Down syndrome that develop ALL have CRLF2 rearrangements compared to less than 10% in the non-Down population. We also see poorer outcomes in this population. So children with Down syndrome that develop ALL are more likely to relapse, and they're more likely to have treatment-related toxicities and associated mortality. So our research is really trying to understand why children with ALL, or why Down syndrome is such a strong predictor of ALL risk. And we wondered if there are specific germline variants associated with ALL in children with Down syndrome, and how these germline and somatic variants could interact within the context of trisomy 21. So fortunately, there's quite a bit we do know about the genetic architecture of ALL, as demonstrated in a range of genome-wide association studies. But the one thing I'd like to draw your attention to in this table is that there are currently, or at least among those top hits that have been identified, there are no variants on chromosome 21. So the idea of a gene dosage effect in chromosome 21 related to ALL is not something that's really borne out, at least with our preliminary understanding and data. So we worked together through COG as well as other populations to conduct a genome-wide association study of ALL susceptibility in children with Down syndrome. And we're able to assemble multiple cohorts to this end. And through that effort, we were able to collect genomic information on a little over 500 children with Down syndrome associated with ALL, and more than 1,000 children with Down syndrome that did not have a report of ALL. And these are results from that publication that was highlighted in Blood about 3 years ago now, But I want to draw your attention to three things in this this particular slide. The first of which is that even with a little over 500 cases, we were able to see genome-wide signals. And I think that's notable for those of us who work in genome-wide association studies to see such strong signals. And I think that points to looking at cancer risk in these highly susceptible populations can really yield insights that you may not get in larger populations. The second thing that I want you to note is that several of these effect sizes are relatively strong. Again, maybe not surprisingly. And even though our top hit was in IKZF1, one of our strongest associations observed were in this missense variant in CDKN2A, whereby children with these variants were almost 4 times more likely to develop ALL compared to children without. The next thing I'd like to note is that all of these variants have actually been identified in genome-wide association studies of acute lymphoblastic leukemia in the general population. So while we did see these significant signals, they weren't necessarily unique to Down syndrome. And then the last thing I'd like to point out is that obviously there's an absence of variants on chromosome 21. And that's largely due to the limitations of the SNP array data in trying to understand genetic risk related to those variants in the context of a genome-wide association study. So I think something that's setting us up for the next step in our whole genome sequencing assessments that I'm going to highlight here in a minute. So a couple of observations that I think are key that have really led us to the Kids First program and trying to understand this risk better is that what we did see is that when comparing these known risk variants in Down syndrome associated ALL with kids that did not have Down syndrome, but also developed ALL, is that there were variable allele frequencies even across these populations, whereby certain variants like CDKN2A seem to be more frequent among children with Down syndrome and ALL compared to children without Down syndrome who developed ALL. Whereas other variants like ARID5B were seemingly less frequent. So what's beginning to emerge, I think, from this work is that there's a variable penetrance or that the penetrance of these variants does seem to interact with trisomy 21. And again, our finding on chromosome 9 related to CDKN2A, I think is particularly notable, and that no matter the ALL phenotype or ALL subtype that was evaluated, we saw a higher frequency of these risk variants in a Down syndrome population compared to a non-Down syndrome population. So suggesting that some of these risk variants are perhaps more frequent, especially in the background of trisomy 21. So setting these children up for a greater risk of ALL susceptibility than a non-Down population. So conclusions from the GWAS, again, that have really led to what we're planning to do as part of Kids First is that the established non-Down syndrome ALL susceptibility loci do seem to contribute to ALL risk in children with Down syndrome. And rather than exhibiting a unique susceptibility locus, trisomy 21 seems to modify the penetrance of ALL susceptibility. And we often saw a greater magnitude effect in the variants that were identified. So our next step is really to take this cohort that we were able to assemble through our GWAS and include it as part of Kids First, and also through the Include project, which Marcia also alluded to in terms of integrating across NIH efforts. And what's notable is that children with Down syndrome not only have this elevated risk of developing acute lymphoblastic leukemia, but they're also at a greater risk of developing certain structural birth defects. So AVSD, or atrioventricular septal defect, is probably one of the best examples of that, whereby children with Down syndrome have a 2,000-fold increased risk of being born with AVSD compared to children without Down syndrome. So this really sets the stage that risk-associated genetic variants in the background of trisomy 21 could lead to a higher penetrance of multiple co-occurring phenotypes in children with Down syndrome. And this includes AVSD and acute lymphoblastic leukemia. And this was the foundation of our XO1, where our two aims are really focused first on identifying genetic variants underlying AVSD in children with Down syndrome, and then taking that larger population and trying to identify variants that underlie ALL risk in children with Down syndrome, with a particular focus on rare structural and chromosome 21 variants, which are mechanisms that we weren't able to fully assess in our GWAS. And in terms of resources with the DS-ALL sequencing, what I think is a real advantage is that we could include not only germline samples, but also tumor samples on these children, whereby we have a little over 350 paired samples to look at germline somatic interactions in this context. So the studies that are underway are really really pointed to doing a comprehensive analysis of heritable variation associated with ALL risk in children with Down syndrome. And as I mentioned, we really want to focus this study on structural rare and variants in chromosome 21. And then the next step, I think, because children with Down syndrome have co-occurring birth defects, is to try to identify the role of co-occurring birth defects in this population on ALL susceptibility. So we do have some primary or preliminary data from our registry linkages that children with Down syndrome that have multiple co-occurring birth defects are more likely to develop acute lymphoblastic leukemia compared to those with fewer birth defects, and that there are specific birth defects that may be predictive of ALL risk. And then linking those children in COG to other relevant data sources to understand outcomes, including early mortality. So I think this is a particularly important area of study because children with Down syndrome and ALL have this distinctive clinical profile that really does predict worse outcomes. So trying to understand the biology that underlies these associations, I think associations are going to be key for better risk stratification as well as treatment strategies. And while Down syndrome does not yet seem to have a unique loci that predispose children to ALL, that they do seem to have a different penetrance pattern whereby these variants do seem to have stronger effects. And I do think it's going to be important, again, through the Kids First program to identify these rare structural and chromosome 21 variants that we can assess through our whole genome sequencing data to understand what proportion of ALL risk is associated with these mechanisms. So I hope that, as part of this today, I've really tried to highlight why Kids First provides, I think, a unique opportunity to explore the overlap between these two phenotypes. And I think related to that, it's easy to see that some of these associations really could be driven by uncharacterized syndromes. And there have been a number of cohorts included in Kids First data that can be leveraged to explore this possibility more than any one group could really tackle. And I think insights into these factors influencing cancer among children with birth defects have multiple implications in terms of how we treat and guide and counsel these individuals. And again, this could lead to improved genetic counseling after genetic testing, surveillance strategies among children with birth defects, as well as treatment interventions. And this effort has just been an amazing collaboration, I think, with individuals that are really countless. And these are a few individuals that have been key to some of the data that I presented today, but the number of individuals I think would go on a presentation much longer than I would have time to present. And with that, I'll end my presentation and thank the organizers again for inviting me to present our work today. And I think that questions and answers are going to go after this session. Is that right, Marcia?
That's right, Philip. We will ask the audience to add their questions to the chat, and we will be addressing your questions in the end of the program. And so let me now introduce you guys the next speakers. Share here with you. I love to call Ada Resnick and David Higgins to the podium. Thank you guys for being here today. Dr. Adam is the co-director of Data-Driven Discovery and Biomedicine at the Children's Hospital of Philadelphia. He is a principal investigator of the Kids First Data Resource Center. And Adam's area of research is cell signaling mechanisms and oncogenes and tumor progression in brain tumors. Dr. David Higgins, he is the program manager at the Kids First Data Resource Center. And his areas of research is bioinformatics and genetic variants. With that, I will pass the word to you both. Adam and David will talk about the Kids First variant search tool, exploring data sets to jumpstart analysis. Thank you both, and I will pass the word to you guys.
Thanks so much, Marcia.
Yeah, thanks so much, Marcia. And while David pulls up the presentation, I'll only introduce for about 5 minutes some of the framing for the Kids First data resources and where we are, and then turn over to David for the remaining of the presentation. But it's always a thrill to follow up in presenting after Philip, in part because Philip is just an exemplar of that hypothesis that Marcia set forth that is the underpinning for the Kids First program, namely that through the intersection from a developmental perspective across pediatric cancers and structural birth defects, we can drive discovery, greater understanding in ways that might be both preventative and therapeutic in nature, and drive more discovery overall around mechanisms, basic mechanisms of disease. And as Marcia highlighted, the data resources themselves, the data sets, have really exponentially grown to a scale that required the Data Resource Center to continuously innovate around how the platform can best support these large-scale data. But importantly, and this is the part that David will highlight, the challenge for us is to follow up on a second hypothesis of the program, right? So if the first hypothesis is pediatric cancer and structural birth defect can intersect and synergize on behalf of discovery and the advancement of care, a second hypothesis is that in order to support that, we can innovate on the way research is performed through new innovations and technology that really highlight the opportunities for that discovery, not only for the EXO-1 investigators who are submitting samples and generating data, but for the research community at large. Next slide. And so really the framing here is, or the question at hand for us as a DRC, is to continuously, in an iterative fashion, innovate around the way that annotated, clinically annotated or phenotypically annotated biosamples that drive new data generation can end up at the impact phase of research, and transforming that data into harmonized information through reproducible resources that are provisioned by the DRC in ways that can drive the knowledge creation process in the way that Philip highlighted, and ultimately change the course of disease or care for children with these disorders or diseases. And to do that, we continuously have to iterate and deploy alongside these data sets new technologies within the platform in ways that address both the size and complexity of the data. Next slide. And we try to do this in a way that supports discovery across diseases in the way that Philip just highlighted. Next slide, across modalities, across disciplines, and then across data resources, which is a phase we're moving into that Marcia just touched. And it's really thinking about that cross-disciplinary framework that we continuously try to deploy new resources that we hope you as a community engage, provide feedback, and we continuously iterate on that process with the goal of accelerating the discovery process across, a platform-empowered resource that brings together communities on behalf of a framework of discovery. And so we're really at that phase where sufficient data and enough resources have been accumulated within the Kids First ERC in ways that moves us into a framework where not only are the EXO-1 investigators who have been provided the resources of sequencing are now leveraging that data for discovery, but where we hope the community at large comes together and drive this forward. And so that can happen both through the portal interface, as David will show you, and then one of the really exciting frameworks that I think we are excited to participate in is how these types of resources within the Kids First, not only at the data layer interconnect with other data sets, but at the platform layer begin to interconnect across other programs across NIH that essentially allow connectivity between data sets that reside across different settings within the NIH, but can be still empowered within a common computational environment. And I think that's really an area of innovation that we hope to drive, and you'll see that as part of David's presentation, that cohort building activities that begin within Kids First can then further downstream intersect within a computational environment that supports the intersection of data sets from various sources, including you yourself as an investigator with access to those resources in ways that's newly empowered through some initiatives that you'll hear about a little bit later in the program. These resources are part of a complex environment that David just highlighted and Marcia highlighted. And so this complex environment is still evolving, still maturing, but it's a rich and fertile area of innovation that we hope any of you who are participating in any one of these efforts or others continuously interacts with us within the Kids First ERC to continuously inform how we might do better and continuously improve on these resources on behalf of your efforts as investigators and community stakeholders within the pediatric landscape. And I'll turn it over here to David, who will go through a use case framework that really starts trying to leverage, the more than, I think, 300 million variants that are currently available for exploration across more than, I think, 60 billion occurrences across these data sets to drive a process of discovery as a model. Back to you, David.
Thanks so much, Adam. Yeah, so thanks for that great introduction. So today I'm going to talk a little bit about the variant search tool. So this is a feature that the Kids First DRC launched on the portal last year that we think is really a great opportunity for folks to be able to compare and search the genetic data across Kids First data sets and able to find these cross-disease comparisons and make these discoveries that we're looking to drive forward. I do want to squeeze in my acknowledgement slide right here at the beginning. So the Kids First DRC, Adam and I are standing here presenting today, but we are a huge group of individuals. And the variant search tool was built by our partners at St. Justine under the team of Dr. Vincent Ferretti. So just wanted to acknowledge them for all their great work putting together this really exciting tool that I'm going to show you today. So what is the variant search tool? So it allows users to be able to search the Kids First genetic data. And so we already have the Explore data tool, which allows users to be able to explore based on phenotypic and clinical data. And we have the file repository that allows users to be able to explore files based on different file types. And so variant search allows you to be able to search the genetic data. And so what you can then do is then you can use this to identify new samples and build cross-disease cohorts. Which goes back to our title, which we hope will allow users to be able to jumpstart these discoveries in pediatric cancer and structural birth defects. So when you go to the portal, you'll have this is what our tool looks like. And I'm going to show some of the features here, and then we're going to go into some use cases where we walk through how could we use variant search when we dive in here in a minute. So when you first log into the portal, we have a main menu up at the top. And you can navigate to the search tool by clicking that variant button right between Explore Data and the file repo. Once you click that button, it'll take you to this page. And so over on the left-hand side, there's a column that you can use to apply filters to the database. So you can imagine, as Adam said, we have millions and millions of variants here. And so you can filter down to find specific subset that might be relevant to your research. And so you can apply those filters. Once you do, those filters will appear in this query up at the top of the tool. So here we're looking for variants that are in the gene MYC, and we're screening for where they've been VEP annotated to have a high or moderate likelihood of altering the protein's function. And then below, the results will be shown. And so here is each of the individual variants. So at the particular locus on the chromosome, links out to dbSNP if available, and spells out protein consequences in addition to showing which studies and how many participants that variant is found in. And so to show you some of the features up close, I wanted to pick some examples. And so I actually wanted to build on some recent discoveries using Kids First data to be able to explore and dive a little bit deeper and begin to make these comparisons. And so first I'll introduce this story about cleft palates, so orofacial clefts. So cleft lip and cleft palate are among the most common birth defects worldwide. And Philip did a great job. He showed some GWAS studies, which can be used to identify variations in individuals associated with a particular phenotype. And so, of course, these GWAS studies can also be done on individuals with orofacial clefts as well. And so a study in 2020 from Dr. Mary Marizita's group did a GWAS using Gabriella Miller Kids First sequencing data. And their group identified a region on chromosome 21 in a population of participants on patients from Colombia that were associated with orofacial cleft. And so we could then imagine this question, well, is this variant present in participants in other Kids First studies as well? So I'm going to exit out of my slideshow here so we can actually search for this variant in the variant search tool. So I'm going to copy that RSID and I'm going to jump over to the portal. And so here I am in the portal and I'm over in the variant tab in the variant search. And so we can search, this is a variant. So we're going to use the variant set of filters here. And then I'm going to paste that RSID and there it pops up. And there appears in the tool. And so it tells us information, yep, this is on chromosome 21. It can link out to the dbSNP page to learn more about the particular allele. This one is in an intron, it's not affecting the protein. And it tells us some information about the number of studies as well. I can click and learn more about this particular variant. If I show different frequencies, and these are, as variant is actually quite common, it shows them in many Kids First studies. And so it indicates these are our different Kids First studies, and this is the frequency of how many participants in that particular study have this variant. So if I go back to my page here, and so we said this is very, very common. So 5,000 different participants have this variant in their genome as we identified through the Kids First DRC harmonized data analysis. So I can actually click this 5,000 participants and it will take me over to the Explore data tool. So now I have a query here where I have, this is the 5,324 participants. And then I can continue to kind of add additional filters to explore this set of participants further. So you can see, for example, the most frequent diagnoses here is cleft lip, cleft or palate, but they also have other diagnoses as well across cancers and other birth defects. So if I look a little bit deeper, I could, for example, narrow this by study. So I could say I'm interested in the kids in the heart defect study. And then I can narrow that further. And so now my frequent diagnoses include various heart defects phenotypes. But I can actually add back, but I'm interested in maybe the kids who have that orofacial cleft. So I can go into the Observe Phenotype, our HPO browser, and type in orofacial cleft, use this term right here and add it to our query and apply. And so we get to this very, very specific query here. So this is participants with this variant in the heart defect study who also have the orofacial cleft phenotype, and there's 2 participants. and we can learn additional information about them below. And from here, then I could take these two participants, explore, what files are available for them. And from here, if I'm interested in exploring, for example, the variants, I could select the GBCF files, and then I could move these files over to Keynote for analysis. And so I'm going to come back to these in a little bit at the end of my talk, but showing how you can use the variant search tool to identify participants, find the participants with that variant, and then move them to Keynote for analysis. So to jump back to my slides, so that's a single variant. So we looked up one particular variant in the variant search. But we can look more broadly than that as well. And so we can look at, for example, some findings from the neuroblastoma study. So neuroblasts are a type of cell found in unborn children, which will develop into neurons. In some children, neuroblasts continue to grow and form tumors, and that cancer type is called neuroblastoma. So neuroblastomas are frequently arise in adrenal glands, but can be found in other tissues as well. And so a study in 2019 from Dr. Sharon Diskin's group found that a small deletion on chromosome 16 was found to be more frequently observed in patients with neuroblastoma. And so using Kids First sequencing data, they helped support that finding by looking at coverage in our trio-based studies here. So here's mom and dad, and then it's actually a de novo deletion in the patient where you can see coverage across that position in the genome is lower. And so this region of chromosome 16 encodes many genes and potentially many variants. So could we do a larger search? Could we search across this entire region of a chromosome and see what variants in that region are present in other Kids First studies? And so yes, we can. So I'm going to go back to our variant search here. And I can even save that query, and I'm just going to do a new query right here. And so we can go back to that a little bit later if I want to pick that up again. So I can search based on by variant. And so we can search based on chromosome. And so this is chromosome 16. So we can select that. And so now we have filtered our variants just to those on chromosome 16. And then I can filter based on our chromosome coordinates as well. So if I put in my genome coordinates, copy and paste right there, and click apply. So now you can see we're looking at chromosome 16, and we're only looking, within that framework along the length of the chromosome. And so what this pulls up, you can see, wow, there's quite a lot of variants. So there's over 700,000 variants. So this could be, a lot to sort through. So we can actually filter this further. So we can say, well, maybe let's just look at the ones that have a high VEP score. So are most likely to be affecting protein function. So we can add that filter, and that gets us down to 785 variants. And so from here, we can then scroll through. So because we're looking at multiple variants at once, these are going to be present in multiple genes that are present in that region along chromosome 16 as identified by that study. So these are rarer variants. And so these are only showing up in a very small percentage or a very small fraction of participants. But you can learn which studies these particular variants are arising in. So if we go over to frequency, this gives us information that these are the 3 different studies with the number of participants that have that variant. And so then an investigator could apply for these studies to receive access and then be able to access that data in the Explore data tool and also push it to the file repo and to Keynote. Lastly, I'll show you, so it can be used to look up existing findings, but it can also be used to make discoveries as well. So we've loaded a lot of existing annotation information into the database as well. So here I'm showing 2 types of birth defects. So on the left, that's an atrial septal defect. And so this is a hole or an opening in the atria that allows blood to flow from the left side to the right side. And on the right here is a congenital diaphragmatic hernia. This is a hole in the diaphragm that allows organs such as the liver to develop out of place. So both of these are birth defects, and so we can ask, is there an overlap in genes underlying these birth defects? And are there any variants in these genes present in Kids First participants? And so we can answer this first question, are there overlap in genes underlying these birth defects? Because we've loaded annotation information into the tool. So under our pathogenicity tab, if I do a new query, under our gene tab, I can search under HPO. And so this will pull up, so this has our human phenotype ontologies. This is genes that are known to be associated with these particular phenotypes. And so if I search for atrial septal defect, I can apply. So this tells us all the variants in all of the genes in Kids First that are associated with this particular phenotype. Then I can add a second filter where I'm going to look for our diaphragmatic hernia, and we're going to apply that. And so now this is telling us two separate sets of variants, and you can imagine I want to do like a Venn diagram overlap there. So I want to compare these two and combine these queries together. And so yes, there is overlap. And so for example, this gene SM3 is implicated in both septal defects and diaphragmatic hernias. And it gives us information about what studies and how many participants in these Kids First studies have these variants as well. So it allows you to be able to look up and continue that discovery. So that's variant search, and so it allows you to be able to search and identify variants across Kids First data sets. And then once you make that discovery, as I showed, you can push those files from the file repo over to Keynote, which looks like it logged me out while I was inactive watching the earlier presentation. But if I log back in really quickly.
Maybe while you're logging in, David, I'll just make a comment. The framework within which David is operating right now is a framework that's accessible to anyone, right? So anybody can log into the Kids First portal into what we term the registered tier framework, right? That once you go through a click-through agreement, you can have access to the clinical phenotypic data and then to this information in the variant browser, as long as it meets certain criteria. So you'll see, for example, in the view that David is highlighting now, the studies numbers are linkable, and you can click on them and explore that data. But if you have below a certain number of participants, you will not be able to click on those and go directly, right? So here you have 2 participants, and so without having dbGaP access, you won't be able to actually view those. And so we're really actually, really straddling that informational space where we are trying to allow investigators to explore across a large number of studies in order to assess what studies they might be interested in to take a deeper dive into in the format that David will just show you, right? So once an investigator has access to such data through dbGaP, once they have done the cohort creation exploration, they can operate across that data and other data through Keynote, which is where David is going to head to next.
Yeah. So these are just to show, so these are my demo project here for the webinar. So these are the VCFs, and so we actually have the indexes that were provided as well. So you can move files from the portal to Keynote, and then they appear here. And so from here, Keynote has a couple different features. So if you're doing a large scale analysis, you can build your own app and workflow. But today I'm going to show off the interactive analysis, which lets you be able to access these files here in the cloud. And so, for example, I'll show that it has for example, a command line interface where you can interact and see, yep, there's my VCF files. And so you can review them at the command line and run your any Unix-based tools. But you can also run notebooks as well. And so here, for example, this is actually a demonstration from a talk by the NIH data scholar Andelette Mays, where we're loading data. So you can load R packages, and then by loading your data set, we can then run our command and then begin to do plotting and analysis. This is demonstration data. I didn't want to show the controlled access VCF data today, but it gives you a sense of, you can go very quickly from variant search to the file repo, to the files, and then you can run your analysis on the VCF and begin your, your graphical output very, very rapidly, all in the cloud. So I see we are approaching the end here, so maybe I'll give one last shout out here at the very end. So thanks so much for giving us your attention today. If you want to learn more about Kids First platforms and data sets, we have a monthly office hours series. And so our these are usually on the second Tuesday of each month, but that was yesterday. And so we wanted to move it so that way we could give you all an opportunity to join. And so we've rescheduled the May session to next Tuesday, Tuesday, May 17th at 3 o'clock Eastern. And so if you're interested in joining, there's a link there, as well as a link in the chat. And we're happy to follow up and give more information about how to use variant search, how to use Kids First data sets. And we're happy to continue the conversation after the webinar ends today.
Thanks so much, David. Thank you, everybody.
Thanks, everybody.
Thank you, Adam and David. Really exciting, everything you showed. And amazingly, we are on time. So thank you so much, all speakers, to keep us on track. Now I'm gonna introduce the next speaker. And I see Valerie already joined. So for those of you that don't know, Valerie Cotton used to be Kids First program manager, so it's humbling to have her here, and is still being part of the Kids First program. So she's still actively, I'm not gonna let her go too far away. Valerie is the Deputy Director of the Office of Data Science and Sharing at the NIH. And as I said, former program manager at the Kids First. And the areas of interest, she continued involved in the program policy, data sharing, and interoperability. With that, I will pass the word to Valerie.
Awesome. Thank you, Marcia. That's right. Nobody's getting rid of me yet. Can you all see my screen now?
Yes.
Awesome. So I am so pleased to talk about Kids First Cloud Credits, and this is a new opportunity that we're offering to all researchers. But I'm going to give a lot of background in history and really frame why this is so important to Kids First. So I think you've already seen versions of this slide today. We share it every webinar. You'll probably see it at least one more time, but I wanted to include it just to emphasize this point about fostering collaboration to accelerate pediatric research. And although you've heard a lot about the Kids First sequencing centers and the DRC and the EXO-1 investigators who contribute data sets to Kids First, and these researchers really all serve as the foundation of the Kids First program, but the program is constantly growing, and it's growing through and even beyond the collaborations with these foundational groups. And as we've built out the Kids First ecosystem, I really love that Marcia introduced this idea of an ecosystem, because that's how we've always thought about it. It's always been a key principle for us that this is not a closed consortium, and that we create space and opportunities for additional researchers and community members to benefit from and contribute to Kids First. And we always get the question, how do I join Kids First? And the answer is always, welcome to Kids First. You're in and you're part of this, and anyone can contribute new tools and knowledge as part of this Kids First community, which of course leads to even more collaboration and the enhancement of other researchers' use of the data. And in fact, this was one of the primary motivations for the Kids First R03 for expert-driven small projects to strengthen Gabriella Miller Kids First discovery. So we specifically designed this opportunity, which is still open. I think James will emphasize that applications for the second cycle are due June 10th. And it's really a place where you can contribute your own expertise to improving the Kids First ecosystem that we have built, even if you haven't been funded by Kids First before. And by building the Kids First data resource as a fully cloud-based system, we're really able to realize this principle. We're able to bring multiple tools to the data that live in the cloud, rather than keeping everything in silos or duplicating efforts in local or on-premises environments. And from a researcher's perspective, a cloud-based approach addresses several key bottlenecks that are usually experienced in a local environment. And I also borrowed this slide from our data scholar, Anne Delatne's, and it really highlights the importance of the cloud. But I updated it to essentially reflect that the future is now. Now we're in the cloud, and now we're offering a cloud credit program that is open to all researchers who are using Kids First data, so that more researchers will be able to reap the benefits of the Kids First data resource as a cloud-based system, which means that you get to use a cloud compute platform like Keynote and community-based standards like standard workflow languages to analyze the data where it lives. And not only is this approach more sustainable for us at NIH, with 48,000 Kids First genomes that are either already released or in the sequencing pipeline, it's becoming essentially impossible to perform this scale of analysis locally. And before I tell you about our new cloud program, I want to share some lessons learned from the internal cloud credit pilots that we've been running internally for the last few years. So these pilots were open to our XO1 investigators, and we received some useful feedback, both in terms of positive experiences and also challenges that helped us shape this new program. In terms of some of the benefits, overall, the cloud program helped lower that barrier to entry to using the cloud, especially for those who had never used the cloud before. And because our cloud credits are allocated through the NIH Strides program and then dispersed through billing groups that are managed through the 7 Bridges software, we're able to be quick and agile in approving and allocating credits for a given researcher for a proposed project. So administratively, it takes us typically 4 weeks on the NIH working group side to review and approve a request, and it takes a matter of minutes to establish the billing group and a Keynote after that, which of course is much quicker than the life of a typical grant cycle. The credits also provided a little bit of a safety net, giving a researcher the flexibility to get comfortable using the cloud and specifically the Keynote platform without dipping into other resources. The cloud credits were often sufficient to help researchers generate preliminary results that could result in strengthening a new grant proposal. And this is actually really the goal here. Use the cloud credits to get comfortable with the environment and plan your next steps for a more robust analysis. And then you can go apply for more funds to continue that work. We heard repeatedly that the scalability of using Keynote is part of what made the cloud credit pilot so enticing. Some researchers commented that in their local infrastructure, they'd be waiting in long queues. They could only run several jobs at a time. But in Keynote, you could run dozens of jobs at a time. You can even configure it so that once one set of jobs finishes, automatically you can trigger the next job. And this really helps you do that really large scale analysis. An extremely valuable outcome is that several researchers use their cloud credits not only to take advantage of existing workflows and pipelines that are already available, but also to build new pipelines that could then be containerized and shared back out to the public for other researchers to use and adapt for their research. You might remember Owen Hershey, who is part of Sharon Plon's XO1 team studying cancer susceptibility, did precisely that. And we had him talk about it this was his structural variation analysis during the 2020 Kids First Fall webinar. So if you'd like to go back and watch that webinar, you can see Owen talk about his approach. He also more recently presented at the NIH Cloud Platforms Interoperability Workshop this past fall, where he described how he is expanding his work to long-read structural variant analysis across multiple platforms using multiple workflow standards. So Owen really is a great example of what the value of these credits could be. Several researchers also took advantage of the ability to collaborate across multiple parties to co-analyze multiple data sets. But honestly, this was also identified as somewhat of a challenge because the security and governance features of the Kids First data resource. For example, making sure that you have the right dbGaP approvals in place across multiple institutions, if there's a lot of researchers collaborating together, needs to be well established, because Kavatica and the Kids First data resource know what the permissions are. They know when you should or should not have access to a given dbGaP data access request. And we'll talk a little bit about how we address some of this later. And as Marcia mentioned, and last year we were able to announce that the NIH Cloud Platform's interoperability collaboration has successfully demonstrated that researchers can now co-analyze Kids First data with data from Biodata Catalyst, Anvil, and the Cancer Research Data Commons. And you can do this all in one analysis environment. So this is a Kavatica-centric point of view. A user chose to start their analysis in Kavatica, and Kavatica knows how to enable the researcher to bring together these multiple data sets. Oops, sorry. And to get you thinking creatively about the types of projects that you could do with your cloud credits, I want to tell you a bit about an interoperability project that we are actively working on. So many of you may be familiar with the Undiagnosed Disease Network. This data set includes over 4,000 genomes, and a large portion of those participants are actually children. And the data are currently available in dbGaP and the Sequence Read archive. And you could use both the Kids First and UDN data sets together. So let's say there was a child represented in the UDN who had phenotypes that are similar to children represented in Kids First. Could you potentially use the Kids First data to better understand the condition from the UDN? And David and Adam, very nicely demonstrated that you have the ability to take the phenotype from the UDN data set and compare it to the phenotypes using the Kids First data resource portal, and look. And additionally, what makes this easier is that we both use a similar ontology. We both use human phenotype ontology. So it's easy for a researcher to semantically compare how similar the phenotypes might be. And then we also both use a technology health IT standard called FHIR to enable that phenotypic data to be exchanged across systems. So after you do that phenotypic comparison, you could then pull together genomic data from the UDN data set and the Kids First data resource into one private analysis workspace in Kavatica. And what all the technology terms that you see in the slide are really doing on the back end is it will let you do this without having to duplicate the data, without having to download it. You're just staying in the cloud. And then with your cloud credits, you can use those funds to focus on analyzing the data without having to pay to store it again, for example. So this is not fully launched. I broke my own rule about talking about something that's not done, but I just think it's a really good example of the type of creative project you could propose to cross-analyze Kids First data with other data sets that are or will be interoperable in the system. We also heard about some challenges throughout the cloud credit pilot. And just to go over these briefly, so I just told you about how when you're working in the cloud, you're working in Kavatica, you don't actually have to pay for the storage. But sometimes there are storage costs. So if you're working on an analysis and you're generating output files, or you've chosen to upload files that you have locally into a Kavatica workspace, that does begin to accrue costs. And in our early cloud credit pilots, in some cases, this led to overspending of the billing groups. Now, we've put some rules in place to prevent this from happening moving forward, which was a requirement to be able to move forward. So now if that starts happening, we have the ability to warn you about it and then shut down or delete data that's accrued overspending in an account. We also heard that researchers just sometimes didn't know how to get started, felt really anxious about how much it would cost. We often hear my favorite tool is not yet in the cloud and it needs to be ported there. And we also observed that not all users really fully appreciated this aspect of value and scalability. And this makes sense because we have a lot of data that's available now, but over the years, it took time to get to where we are now, which is that there are more than 20 data sets available, but researchers were often focused on one data set at a time, and that's changing now that so much data is available. So to address these bottlenecks, NIH staff worked with the Kids First DRC. And I just want to make sure, can you all see my browser now? Okay, I'm going to assume yes, there might be a little bit of awesome.
Yes, you
Okay.
can.
So, thank you to the DRC for working with us to create resources to just facilitate that onboarding. Here's how you can get started with working in the cloud. And this is now live in GitHub, and we'll definitely share the link, and we'll share all the slides. But just to walk you through this so that it'll be easy for you to get started, there's some introductory information about what the Kids First program is, what the Kids First data resource is, how we're in the cloud and why we're in the cloud, and why that's important for you in terms of answering some of the scientific questions that you might be interested in. And David talked about the data sets that are available, and you can even, anytime from your home, you can watch David give a really wonderful overview of how to go from the portal to accessing Kids First data in Kavatica. And that's exactly what the second page is. Walks you step by step with these really nice visualizations about logging into the portal, doing your query in the portal to identify the data set you might be interested in, how to export or access that data, and then of course some information about which data do require that dbGaP approval because they are controlled access, in which you can find in the registered tier. And this information about dbGaP is really important because it gets at what I was talking about earlier. You have to have the right dbGaP data access requests in place in order to do a collaborative analysis across multiple data sets. So this link right here, as well as this video, will walk you through that. We talked a bit about how people just didn't, it's really hard. Where do you begin with estimating what it might cost to use the cloud? And like I said, you don't have to worry about storage of our data, but you do have to be aware of storage of files throughout the life of your project, because it's inevitable to happen that as you work through your analysis, you're generating data products. And this includes some tips about keeping your workspace clean, so deleting files when you no longer need them so that you're not spending those costs. And then some benchmarks of how much it costs to run some of the standard workflows that the DRC has created. So in most cases, you won't actually have to run these on Kids First data. You might have to run it on another data set if you're doing a combined analysis, but it does give you a feel for the type of workflows and how much they cost. And the team also created this cloud cost estimator. I'm going to go ahead and flip to this demonstration tab to show you how you can use the blank sheet and input what type of analysis you think you're going to do, and use some of the published AWS costs, which are here on the reference tab. These change sometimes. So, David created this based on the numbers that were available that day, but you can also update the spreadsheet yourself, and then it'll automatically compute for you an estimate of what it might take to do the analysis that you're aiming to do. So let me switch back. How to apply for cloud credits. So this is why we're here today. So it's a really simple process. I'll talk a little bit more about how we're going to handle this on the NIH side. But to get started, you basically send an email to the Kids First mailbox. And to get started, we'll ask for a few pieces of information, but then we'll respond with next steps. So we're going to need a little bit more, depending on where you are and what kinds of data sets and analyses you're planning on doing. Managing cloud compute costs, again, some strategies for not overspending, keeping your workspace clean, being aware of how your billing group is being spent. And then typical Kavatica, these have always been available, but we tried to put them all in one place for you to get started. And then of course, closing out your cloud project. And, again, after you've used cloud credits that have been allocated from NIH, we really encourage you to continue using the cloud, but you might have to commit resources from an NIH grant or somewhere else to do that. So with that, just a summary of what this will look like on the NIH side. So I also have a million people to thank, but I definitely want to thank Wei-Ching Lee, who really spearheaded the development of this process. She's been chairing the review committee, who has overseen the review of the cloud credit pilots for the last several years, and has graciously agreed to continue even as we move forward with opening this to everyone. Generally, the process will be that to get started, you could ask for up to $1,000. If you're also doing a cross data set analysis, so that interoperability use case of co-analyzing data with Anvil or Biodata Catalyst or Cancer Research Data Commons, you describe that and you could be eligible for another $1,000. After that initial work is done, we'll ask you to tell us how it went before you can apply for more credits, but you can apply for up to 5,000 more credits after that initial allocation. And then you could even apply for even more after you share workflows or pipelines. So the kind of work that Owen did with using his credits to fine tune and then build a new pipeline and share that back out with other researchers. We have some administrative procedures in place. If we create billing groups, but they go inactive for 6 months or more, we will shut them down. And we'll also take steps to delete workspaces that are overspending. And then I've said this several times, but you can continue working in the cloud even after the cloud credits have been allocated. So this is my last slide. We will be announcing more broadly that we're essentially open for business, that researchers can apply for cloud credits. But these are the basic first steps. And that is my last slide. Thank you all so much.
Thank you, Valerie. And we now will, I would like to introduce you our last speaker. Just give me a moment.
My computer is exceptionally slow today.
It's a great pleasure to have here today James Coulombe. James is the Chief of Development Biology and Structural Variation Branch at the National Institute of Child Health and Human Development at the NIH. And he is the Kids First Program Working Group Coordinator. James' areas of research is human development and signaling pathways. And James, it's a pleasure to have you join us.
Let me see if I can share my screen.
And while you were doing that, I want to ask everybody to, if you have questions for our speakers today, the audience can add your questions to the chat or questions and answer box. And we're going to be addressing your questions right after James finishes his talk.
Sorry, my computer is like moving really slow. So
That's okay.
apologies.
Okay. Thank you, Wells.
It should be there, but
Yes, we can see your slides now.
let's see. Get all this. It's there we go. Man, I don't know what's happening here. Just to remind you, the vision of Kids First is emphasizes collaborative research and data sharing. And to realize this vision, the goal of Kids First has been focused primarily on building a data resource so that the genomics of mostly individually rare conditions of childhood cancer and structural birth defects could be studied. Let me split that away. There we go. I'm sorry it's so slow. Next slide. Wow, this is just amazingly slow. And it's not, there we go. However, there was authorizing legislation, Gabriella Miller Kids First Research Act, that limits how we can spend the money for Kids First. We have to use the money to supplement but not supplant funds that otherwise the NIH would have and be using for pediatric research. We also have to make grants, and we can't do other kinds of mechanisms like contracts and whatnot. So that does limit things a little bit. And yet we've always understood that in order to fully realize the value of the data resource we wanted to build, there needed to be support for the extramural community to do analysis, discovery, and validation using the data. So the first way we kind of, as it were, got around things is to instead use the regular NIH Institute's money to fund R03 applications, either small research grants. Typically, they were $50,000 a year, but we managed to get an expansion of that to $100,000 a year for direct costs. And they can only run for 2 years, so they're good for, in fact, specifically intended for things like secondary analysis of data sets. Preliminary data is not required, and we've been issuing this kind of grant using the individual institutes' monies for the relevant institutes that who are interested in your particular project science. NICHD, NCI, NHLBI, NIAAA, and NIDCR all signed on to these program announcements with referral. And we have funded, well, we received 97 applications so far. We've managed to fund 24 of those, so that's not too bad. They're competing against the individual IC pay lines. And we still have some application dates available coming up in the fall for the next round of these R03s. So that's one mechanism you can use to fund analysis using, and specifically this program announcement requires you to use Kids First data. Let's see. Valerie already told you about this other opportunity we were able to do an RFA. That's a request for applications, and it basically has money set aside to pay these grants. The last round, we were able to fund 11. We received 11 applications, and we were able to fund 6. One of those to the Include program. We have a deadline coming up very shortly on June 10th. If you have a project that you think would fit this funding opportunity announcement, and that would include development of analytical tools, addition of phenotypic or clinical data to existing genomic data sets, reconsenting of subjects where data use limitations might limit the utility of the data, or analysis and curation of existing data sets, especially when you can pitch that so that the work illustrates and enhances the usefulness of the data resource. We're kind of liberal about what we consider to be strengthening Gabriella Miller Kids First discovery. You can ask us whether we think your project would fit, but you know, you don't have much time and need to if you want to apply to these, you need to get on it. And I've already started getting some letters of intent. As for all NIH grant applications, letters of intent are not required, so you don't have to do that. But you really should talk to one of the program officers, me, or you should send an email to Marcia, and we'll get back to you about whether we think your project would fit. Sorry, I'm waiting there. You can always fund your projects through investigator initiated or the so-called parent funding opportunity announcements. Those include R01s, which all institutes honor, R03s, which are a little bit more limited. And in this case, applying to the parent, you would be limited to $50,000 in 2 years, $50,000 of direct costs. And there's also R21 that various institutes use in different ways. Some insist on them being high risk, high reward. Other institutes do not. So you probably should, again, talk to a program officer before you put in an application or go to all the trouble to put it together. I should say that we have seen successfully funded R03s, R01s come through using Kids First data. And I have observed and read in several summary statements for either R03s or fellowship applications where the initial applications didn't fare so well in review and were kind of vague about the source of the data that they were going to be analyzing when the resubmission specified that data was coming from the Kids First Pediatric Research Fund data resource. That seemed to help with the reviews, and they got more favorable reviews because the data was known to be there, existing, and they could point to the Kids First Data Resource Center as the source of their data. Let's see. Oops. I don't know what's happening there. As always, you can go shopping on OER's grants website. And there's a little search function, a really quick search pulled up these 3 opportunities, which would be applicable to Kids First data, even though they don't specify. The first PAR listed there are for R01s. This is actually sort of salting the mine here. This is out of my branch for screening and functional validation of human birth defects, genomic variants, and also NIDCR and ORIP have signed on to this. And this is really for identifying variants and taking them the next step to functionally validate in animal models what the biology of implications of that gene, that variant are. There was also in this little search, quick search is an RFA for tools development, novel tool development from the Cancer Institute, NCI. So that's a good possibility if you're developing tools to use to analyze cancer data. And also an R21 PAR from NHLBI showed up, which is basically for secondary analysis of existing data sets. And that could include Kids First data very easily. So I'll emphasize that you should always get in touch with the scientific contact for any of these funding opportunity announcements to discuss your work. And in general, you should always talk to a program officer about what you'd like to do and let them help you find the most appropriate ways to try and find funding. That's a big part of their job, and generally they're quite good at it. So definitely let us help you, and especially let us help you find ways to fund your work using Kids First data. And from there, that's my presentation. Thank you.
Thank you, James. And thank you again, all speakers today. We have some time for questions, and but before I go to the questions and answers, first I would like to invite all speakers to turn on your cameras, as we have questions for all of you. As you would join, I want to kind of my take from today, and I hope you guys got same take, is that the meantime that researchers can still download the data, they can also now be part of this larger ecosystem of data in the cloud. And as we create an environment with the Kids First program for collaborative research and new discoveries using the Kids First program platform, through the Kids First Data Resource Center. So join the portal, which is not only available to the principal investigators of Kids First program, but also all researchers out there can leverage that large data environment for your own research and discoveries. And also how NIH, and specifically the Kids First program, is using pilots to enhance opportunities for research and discoveries. A good example is the cloud credit pilot, which addressed a question that is a relevant question in the field with concerns of cloud credit computing. And we're addressing that by offering these opportunities for people to test it out, the waters, see how it works for your own research. So these are some of my taking from today's session. And I will start with a couple of questions. I'll start with a question for my own question for Dr. Philip Lupo. I would like to take the opportunity, Philip, as you're here, and you have experience not only with Kids First program, but with your research. I would like to ask your advice for the early career researchers that are out there starting their own path in pediatric cancer research and pediatric research in general, with birth defects, fields, and other pediatric conditions. Your advice to the young people, and also how they can leverage from the Kids First program support.
Yeah, that's a great question, because when I started doing this research, I was young and had less gray hair. In fact, I think I alluded to the fact that I was at that 2012 seminar on the overlap between birth defects and cancer. And I think the one thing I'd like to say in this research is I do think that you have to have a certain amount of patience. You know, if you're just getting started, you know, take the time and effort to help build these collaborations. And you know, you could look at it and say the task is too onerous, but you know, within time, I think you'll find some satisfaction as you draw people together. But then I do think that the number of questions that could be asked right now with Kids First data are beyond my scope. So I can't imagine that there are you know, hundreds of things that young investigators could really dig into with the existing data. And you know, if you can't think of one, feel free to email me, because I'm happy to chat about the kinds of opportunities that I think are there. Like I said, I you know, I wish I had enough time to dig into those 48,000 genomes. Is that what you said, Valerie? But
yeah, it was from the fall, so it's getting bigger every day, right?
I mean, it really, I think so many questions can be asked and answered with these data that it's a resource that you could use right now. And as you can see from the presentations, there are so many grant opportunities that you can take advantage of, either existing program announcements or you know, some of the parent announcements. I think as long as you're leveraging existing data, you know, that's always an advantage from a reviewer and funding agency's point of view. So I hope that answers the question, Marcia.
Thank you, Philip. And this is also a question for Adam. I don't know if Adam wants to add something.
I'll just add that, one of our key hopes, and like Philip, I was once a young investigator, but one of the key hopes is that what's different now than when either Philip were young investigators is that the inertia that is in front of a young investigator to acquire the requisite resources to do analysis is dramatically different now, right? So our hope is that the platform resources that are here essentially democratize opportunity in ways that here you have data, you have compute resources, you have cloud credits environments. So a lot of the barriers to entry in the past for researchers around the need to generate data, acquire resources, local or otherwise, are essentially being solved, hopefully, through this infrastructure in ways that we really hope young investigators latch onto and accelerate both their own career and the discovery process.
Thank you, Adam. And also I have a question. As a cancer researcher, I could not help myself but think my own being able to work with the Data Kids First program, and actually is something I'm planning to do, and I have some ideas. But my question to David is, I'm a cancer researcher, I'm not a bioinformatician. So are there opportunities through the Kids First program, Data Resource Center, for people like me to also explore data and use the data?
I'd say yes, definitely. So we support a number of different tools and opportunities for users to be able to engage in the data across kind of the realm of experience and background and expertise. And so especially since you mentioned you're a cancer researcher, so we support the PEDC Bio portal, which is a website that I didn't show in my demonstration today. But we load each of our Kids First cancer data sets, the somatic data, into the PEDC Bio portal, and that allows users, yeah, there it is, Adam. Thank you for pulling it up for us, to be able to point and click and explore the data in the PEDC Bio portal to be able to access it further. And so you can kind of identify the genetics in tables. It's all point and click. But beyond that, you can also do plotting and comparison and discoveries as well. So all of our Kids First cancer studies, like I said, the somatic data is loaded up in PEDC Bio portal. And if you or any other cancer researchers in the audience want to learn more, this would be something that I'd love to go over at an office hour session or a future one-on-one.
And I couldn't help myself while Philip was presenting, and Philip was spoke about USP9X. I just did a query on the Kids First portal. Moved into the PEDC Bio portal environment and validated that everything Philip said is true, as related to here what you're seeing is really like deep deletion events across a variety of pediatric cancers. What you're also seeing is, in addition to the leukemia cohort that Philip mentioned, you're seeing them across other disease contexts in ways that, hopefully Philip or a young investigator can take on and ask additional questions, about the significance of these findings. So very rapidly, and again, for me, this is a testament to the work of the team on the portal, is when we become users of the portals themselves, even as presenters are presenting data, it's a sign that, and I guess that's my litmus test. If I can do it, I feel like Marcia or anybody else can do it. We're really excited about iterating around this framework, and are always looking for feedback from the non-bioinformatically trained researchers on how we can improve these resources.
Thank you, Adam. And also I want to answer some of the common questions here, or if we're going to make the webinar available, the recording will be available at our website, as well as the slides. However, the office hours, we don't record because we want to maintain the confidentiality of the consultations, as we can provide some deep dive on using your own data. So, but David, do you want to talk about that a little, like in one last 30 seconds, because I want to answer a few other questions, but just to emphasize that the office hours are available, but how people, if they cannot come the next one, is there something you do at regular basis?
Yes, we do. So we do it every month. We do it usually on the second Tuesday. And I've got a link here. I think Whitney posted it earlier in the chat for signing up for our office hours sessions. And so here, I'll paste it right here for everybody. But we cover everything. And so everything from the very beginning of, kids, what is Kids First? How do I get involved? How do I get started? All the way to the very end of, I did this analysis and I have these results, and what does it mean? And everything in between. And so that's my short pitch. And so if however you want to, get involved in Kids First or learn more, it's me, a team of our data analysts and our bioinformaticians, and we're available to speak and answer questions. So we'd love to have you join us. And like we said, our next session is next Tuesday afternoon. I hope to see you there.
Thank you, David, again. And so let's see here. Many of the specific questions have been answered. Thank you, Philip, for answering them directly in the Q&A question that's available to everybody to take a look. You guys can take a look at specific questions, and we will most likely also generate a frequent asked questions during the webinar, and will be available in our website as well after the session today. So we have 1 minute. If there is any other pressing questions, we have still 1 minute to go, but let me take a look here. One question I have here that I would like Valerie to address a little bit is, how can I get access to cloud credits? I think you mentioned a little bit, but maybe it doesn't hurt to just emphasize if someone is interested in access.
Okay, I think the best thing at this point would be to visit that new GitHub page, and we shared it, but I'm finding it myself. Perfect. So this page, you basically start by emailing the Kids First mailbox, and we're asking that you answer a few basic questions, but it's a little bit, the adventure you go on will depend on where you've been. If you are an X01 investigator, or you're somebody who was part of our 03 program and you had cloud credits as part of our internal cloud credit pilot, you may be going on a slightly different adventure than somebody who has never been part of Kids First and is asking for cloud credits for the first time. So I think, from a basic perspective, just answer those basic questions, and Marcia and Wei Ching will be there to walk you through the rest.
Thank you. All righty, so I think we got on the hour. So I will again thank you all the speakers for being here, and everybody that took the time to share this time with us. And I'm looking forward to continue this dialogue with everybody in our next webinar in the fall. Thank you, and see you. Bye.
Bye.
Thanks, everybody.